The term recombination is used in several different contexts in genetics. In reference to heredity, recombination is defined as any process that results in gametes with combination of alleles that were not present in the gametes of a previous generation. So, recombination is classified into two different types. They are interchromosomal recombination, which occurs either through independent assortment of alleles whose loci are on different chromosomes, and second one is intrachromosomal recombination that occurs through crossovers between loci on the same chromosomes. So it is important to remember that in both of these cases, recombination is a process that occurs during meiosis. One thing you have to note is mitotic recombination may also occur in some species, but it is relatively very rare. If meiosis results in recombination, the products are said to have a recombinant genotype. On the other hand, if no recombination occurs during meiosis, the products have their original combinations and are said to have a non-recombinant or parental genotype. Recombination is very important because it contributes to the genetic variation that may be observed between individuals within a population and acted upon by selection to produce evolution. So, recombination is also very, very important for evolution. Without recombination, evolution is not possible. And as an example of interchromosomal recombination, consider loci on two different chromosomes as shown in this figure. We know that if these loci are on different chromosomes, there are no physical connection between them, so they are unlinked and will segregate independently as did Mendel's traits. The segregation depends on the relative orientation of each pair of chromosomes at metaphase. Since the orientation is random and independent of other chromosomes, each of the arrangements is equally possible for two unlinked loci as shown in this figure. More precisely, there is a 50% probability for recombinant genotypes and a 50% probability for parental genotypes within the gametes produced by a meiocyte with unlinked loci. Indeed, if we examined all of the gametes that could be produced by this individual, we would note that approximately 50% of the gametes would be recombinant and 50% would be parental. Recombinant frequency, that is RF, is simply the number of recombinant gametes divided by the total number of gametes. It's already explained in the previous slides. A frequency of approximately 50% recombination is therefore a defining characteristic of unlinked loci. Thus, the greatest recombinant frequency expected is only 50%. In 1964, Robin Holliday proposed a model that accounted for heteroduplex formation and gene conversion during recombination. Although it has been supplanted by the double-stranded break model, at least for recombination in yeast and higher organisms, it is a useful place to start. It illustrates the critical steps of pairing of homologous duplexes, formation of heteroduplex, formation of the recombination join, branch migration, and resolution. The steps in the Holliday model are illustrated in this figure. Here, two homologous chromosomes, each composed of duplex DNA, are paired with similar sequences adjacent to each other. An endonuclease nicks at corresponding regions of homologous strands of the paired duplexes. And this is shown for the strands with the arrow to the right in this figure. DNA ligase seals the nicks. The result is a stable join molecule in which one strand of each parental duplex crosses over into the other duplex. And this X-shaped join is called a holiday intermediate or a chi structure. Branch migration then expands the region of heteroduplex, and the stable join can move along the paired duplexes, feeding in more of each invading strand and extending the region of heteroduplex. The recombination intermediate is then resolved by nicking a strand in each duplex and ligation. Then resolution. Resolution means it can occur in either of two ways. Only one of which results in an exchange of flanking markers after recombination. And the two modes of resolution can be visualized by rotating the duplexes so that no strands cross over each other. In the horizontal mode of resolution, the nicks are made in the same DNA strands that were originally nicked in the parental duplexes. After ligation of the recombination of after ligation of the two ends, this produces two duplex molecules with a patch of heteroduplex, but no recombination at flanking regions. In contrast, for the vertical mode of resolution, the nicks are made in the other strands, that is, those not nicked in the original parental duplexes. You got it? Ligation of these two ends also leaves a patch of heteroduplex, but additionally causes recombination of flanking regions. You should note that horizontal and vertical are just convenient designations for the two modes based on the two-dimensional drawings that we can make. The important distinctions in terms of genetic outcome is whether the resolution steps target the strands initially cleaved or the other strand. Then, the recombinant join proposed by Holliday has been visualized in electron micrographs of recombining DNA duplexes, which is shown in this slide. It has the proposed X shape, although this joint is drawn with some distance between the duplexes in illustrations. In fact, the two duplexes are juxtaposed and only a very few base pairs are broken in the Holliday intermediate. The structure is symmetrical, and it is likely that the choice between horizontal and vertical resolution is a random event by the resolving nucleus. It chooses two strands, but it cannot tell which were initially cleaved and which were not. So I hope you all understood the session. Stay blessed. Thank you.